recorded because it cut out yesterday. So yesterday we started looking at mitosis and cytokinesis. You remember we have the cell cycle. Think about that big ring. These are the two small parts of it. We specifically started looking at what happens as we go through it. There's my pictures here. So where we kind of start is we end up starting here in our first phase, which is basically when the spindle apparatus, these webs start to form. Then in our second phase, our sister chromatids, which are those copies of DNAs, line up in the middle. They're getting ready to split. The third one is our third phase is when they actually start to split. These spindles, these attachments, these hooks start to pull towards each pole of the cell. This is anaphase. And then finally, we're on our final phase where the cell is just about ready to split. It has its own nuclear material now. It's formed the nuclear membrane, etc. And then, of course, cytokinesis is when the cell itself actually splits. So all in all, this is what it looks like, right? You start with a cell. It condenses the chromosomes. It starts to pull it on the equator, pulls it apart, gets ready to separate, forms two new daughter cells here. Then we just ended up going through these questions here. Again, mitosis alone can't produce our cells because it only copies the DNA materials. We have our 4 different phases here, right? PMAT. If you can remember that, great. If you can't remember that, which is fine, think about popsicles melt at temperatures or something like that. You can come up with something better, probably. But that's a way to remember the order that it goes in. So pro, meta, ana, telo. I don't think much needs to know. Oh yeah, and again, it just doubles, like just some basic math there. So that was section 2. So we're finishing off cells today. We're done with cells after today. There will be a reminder and exam for everything cells. Unit 7, Unit 8, Unit 9. That's going to happen tomorrow. Just like the other ones, it is open book. It is on the LMS. The big difference with this one, though, is you get 1 try and it's timed. Whereas the other ones, you've generally gotten some infinite time. So you do want to go in there having some stuff prepared. You want to go in there, maybe having studied, maybe with your flashcards, something. Sorry? Yeah, I got the materials and everything. I recommend it. If you guys ever have a free moment, that's always a good idea. Because every single day, it's here's 5 new things. Here's 5 new things. If you think about it, it's 35 days long. Going to be 200 things you gotta know at the end of it. So it's a really good way to kind of condense that information. Okay, so we're going to finish off here with our last one, which is, let me go to this one here. So again, we know that in mitosis, actually, I don't even want this one. I want a different slide. Here's my other slide. Okay, so again, when we look at the cell cycle here, we know what the cell cycle is. We know the cell cycle is all about its going from 1 cell into 2 cell, right? That's what ends up happening here. Now, what we do know is that in interphase, this is when a DNA starts to replicate, right? This is when it starts to copy and paste that information. This is stuff from the first section, by the way. And if you guys remember, the example I used is, think about if I give you all those books back there. There's like 100 and some books back there. I made you write it all down perfectly, letter for letter. You would make a mistake. It's going to happen. That is a portion, a fraction of what it actually is like for your DNA. It is copying millions and billions of things. How does it do it accurately? We know the consequences of when it is inaccurate. So we're going to look at how this cycle is regulated, because there's 2 problems that emerge. One, which we've already discussed, is, well, what happens if the cell grows and splits and divides and it copies it wrong. You could get birth defects, etc. Like, we talked about that. Can anyone guess what the other issue that might happen when we talk about cell growth? Cancer, absolutely. It's the big C. So like we know that cancer is when cells grow on regular, like tumors are just cells that keep growing and growing and growing and growing. The whole point of chemotherapy, and this is wildly dumbing it down, it's more complicated than that, but is that you're killing these cancer cells that grow quick, really fast, faster than you kill the host of like the human. That's why chemotherapy sucks to go through. Like you're kind of damaging the rest of your body, but it's just like this, the cancer cells get damaged quicker. Is the whole concept. That's how weed killer works, by the way, is the weeds will suck up it faster than your grass. That's why your grass is fine, but the weeds die. Same thing, actually. But anyways, so we know that this regulation is important because either you get the wrong information copied or you can get cancer. You can get unlimited, unregulated growth. Not good. So how does this work? So we're going to look at how something called cyclin proteins work in controlling this. We're going to look at what the heck is cancer. We're going to look at this thing called apoptosis. And then we're also going to look at stem cells. Does anyone know what stem cells are off the top of their head? Yeah, it's unspecialized cells. You can kind of use it in a bunch of different ways. It's not a programmed cell yet. Is this going to be a muscle cell, a bone cell, a skin cell? Is this going to be a cell that could be used for maybe like an important organ, like maybe to help with the whole of the heart or something? So we're looking at a whole bunch of things here to finish off cells. So we have something called cyclins. If you guys are noticing, this thing down here is the cell cycle, right? That's what we just learned about yesterday and the day before, actually. But what if I added to this from yesterday? Just what's different in the picture? Sorry?
The legends.
The legends, yeah. Specifically, we know what these are, stoplights. So that's what these kind of serve as. The whole problem with cancer is it is unregulated. There are ways to regulate the growth, to control it, to say whether it stops or goes. And these cyclins are what do that. So these cyclins are proteins that bind to enzymes we call them CDKs. Basically, what these things do, what these different combinations do, is they control the activities. They're stop and go light. Start, stop, start, stop. That's what they are. So cyclins are stoplights. I'm gonna put stop/go light. I recommend you guys write that down. That is so much easier to remember what something is, right? Yeah, it's the stop and the go light. We know what that is. So when we're thinking about these again, you think of them as a stop and a go light. You can also think about these are the keys for the ignition, right? These aren't what do the cell cycle. These aren't what cause the cell, or these aren't what the mechanism that the cell splits itself. You turning the key in your car isn't what moves the car, it's the engine. But you're telling the engine that it's okay to power on now. So you can think about it as keys in a car, as stop and go lights. Either of those are really fine ways of thinking about this. So these cycles have those built-in checkpoints. These monitor the cycle, and if something's going wrong, abort, cancel, get rid of it. So these cell cycles, the cell cycle has those built-in checkpoints. And so there are checkpoints at each stage of this interface to protect against duplicating DNA damage. Again, it needs to be accurate. Even if it only makes one mistake out of every million genes, that will cause substantial problems. It needs to be more accurate than anything you can really think of, more accurate than computers, etc. So let's look at cancer here. So we know what cancer is. Cancer is when we have uncontrolled growth and division of cells. So cancer is, long story short, it didn't listen to the stoplight. It just keeps growing and growing and dividing and dividing. So cancer is when the cells stop responding to those controls. So, but the big question is, how is cancer itself deadly? Well, things require resources to operate, right? You need calories and water and nutrients and etc. Eventually, the cancer takes more of those, but it takes what's supposed to go to normal cells and it sucks it up for itself, right? And now all of a sudden, there's just not enough food for the rest of your body. There's not enough of this important stuff.
It eats all your.
Yeah, absolutely.
So what you lose weight.
I think that probably could be it. I know you lose weight during the treatment of cancer, and a lot of that is because you're usually pretty nauseous, is my understanding. But I think during cancer itself, yeah, you do lose weight, and that's probably a reason why. Because I mean, it takes a lot of energy to actually grow something, right? Like, like this is, yeah. I mean, it's just growing. So one of the important things here, though, is understanding why itself cancer is deadly. We know cancer is deadly. That's not something we need to write down and be taught, but it's why is it? So the question here is, what causes cancer? Carcinogens. Yeah, we usually call them carcinogens, right? Can anyone list some examples of what are things we know that now are linked to cancer? Yeah, and well, smoking. Yeah, smoking does. Alcohol, radiation. Too much stuff. Bad burns actually are pretty bad. But what causes cancer? Pretty much, it seems like just about everything these days. But specifically, how do those things cause cancer? How does smoking? How does really bad sunburns? How does that actually potentially lead to cancer? What is it doing to your body to do that? It's mutating or changing your DNA. Yeah. So we know that we have those things called carcinogens, right? We know that those cause cancer. The way they do that is they change your DNA and they cause that cell growth cycle and those checkpoints to falter. And again, I'm sure, if we were sat here, we could probably come up with literally 100 examples as what causes it. But the important part here is understanding how it causes it. And it's the fact that your DNA itself gets changed or damaged. Okay, so next is, so what happens if your body detects that, oh, there's an issue with this thing, it needs to terminate it, needs to get rid of it. So there's a process that it uses for that, which is called apoptosis. So this is when it tells the cell to, hey, cell, you just copied the wrong DNA. You need to die now. So what ends up happening is they go through and they shrink and they shrivel. And again, this is really important because a damaged cell with poorly copied DNA, that's what can lead to that cancer. So cancer is either that's damage to the cell process, the checks and balances in the cell cycle, or it is damage to the cell itself. So if it misses even one of these, that's where you can get cancer from. So the nice thing about apoptosis is that's it. It doesn't go any deeper than that. What is apoptosis? The cell shrivels and dies. That's it. There's no specialized process we have to focus on there. It just cuts off nutrients to it and it shrinks, shrivels away, and it dies. It detects the issue, it kills the issue, and it moves on. So going into our final part here is stem cells. So we usually have a pretty good idea what stem cells are. These were things that were quite popular in politics during, I think, the early thousands. They have very useful medical properties, of course, but that's because we know that multicellular organisms have specialized cells. This is the fancy way of saying that we have heart cells, skin cells, muscle cells, liver cells, brain cells, red blood cells, different kinds, right? Most all multicellular organisms have different cells with different purposes. So stem cells are, of course, precursor cells. They are ones that haven't been told what they need to do yet. So you can tell a stem cell, I want you to be a muscle cell, I want you to be a blood cell, whatever it is. So we have two different kinds. We have embryonic stem cells. This is from when you are a fetus. And then there are adult stem cells. And this is something most people don't think about. Most people, if they've heard of stem cells, associate it to fetuses, not necessarily to actual adults. So we'll end up talking about this. With these stem cells, they can be turned into, most things. Not everything. Brain damage is permanent, right? Brain cells, that's it. You got a limited number of them. And there's a couple other exceptions, but generally they can be turned into anything. Your body can tell it to go to a muscle, go to a blood cell, go to a nerve cell, go into something or other, a bone cell, a bunch of stuff, like the word cell, whatever it is. So what happens is, so when we have an egg that gets fertilized, what ends up happening is we have 100 to 150 cells that remain, they're called undifferentiated. AKA, we have 100 to 150 embryonic stem cells. The egg gets fertilized, and there's 100 cells ready to go. What do you need, boss? I'll do whatever you need me to do. So as the development continues, right, this is the 9 months, those cells receive the signals that are going to tell them what to do. So you don't actually have a lot of them in fetuses. So those are the embryonic stem cells, right? What surprises most people, though, is that adults have them. Because most people think of just fetuses with them. So when we look at the adult stem cells, these are found in a bunch of different places, and this can be used to maintain and repair the tissue. But basically, these are a little bit more specialized than embryonic. But they can kind of have different jobs. So what does that mean? Like, what does that mean, more specialized? So maybe it's already been told you are going to be a muscle cell somewhere, but you don't know where. There are different kinds of muscle cells, for example. So it could be something along those lines. It could be you're going to be a blood cell, but maybe not sure what kind of blood cell. It could be you're going to be either a ligament or a tendon, but we're not sure which one yet. So it's a little bit more specialized. So adults do have stem cells, but they are more specialized than the embryonic ones. And they are specifically used with tissue repair. So that's it. That's it for cells. We're done with cells after this. I do have a couple of questions, though, that we're just going to go through. So there are about 7 of them here. So there's a lot, but they don't need a lot of space. I'm going to say, honestly, you only need one line for each of them, except for chapter 4 or section 4. So you just need one line for each of them. There's not a lot. This one, however, is just going to, not 4, is going to need 2 lines, but the rest of them can just be 1. I'll give you a minute. So starting with the first one here, the cyclins. How do these control the cell cycle? Those regulate all parts of the cell cycle. So mitosis, and the 3 parts of the cell cycle. And basically, it starts where it stops. This is the fancy way of saying that it is a stoplight. It is the keys for a car. That's how most people tend to remember this. Is just, it's just the keys. It tells it when to go and when to not go. Okay, so next we want to look at how is a cancer cell different than a normal cell? Basically, it just keeps growing, right? Oh, and then divide. It divides unrestrained. You don't even need that first part. Just it just divides unrestrained. We don't really need to know much about cancer. We have a pretty good concept of it. So next part here was we identify three carcinogens. You guys could do this. Again, these are just some examples. Frankly, you probably don't even have to write this down. Like you guys would be able to do this. Cigarettes, UV radiation, usually sunlight, tanning beds, stuff like that. Asbestos. There's literally hundreds of them. Okay. So what is the difference between apoptosis and cancer? We know that apoptosis is when, we have a cell, it has an issue, it's copied something wrong, we're going to kill it. So it's when it goes under a programmed cell death. But cancer is when this apoptosis is a potential result if this apoptosis does not happen. This is when that cells can grow and they can divide unrestrained as long as they have nutrients. Stem cells. What can we use stem cells for? Basically, they can be used as cure for medical conditions, or they can help with genetic defects. Okay. So what's the difference between an embryonic and an adult stem cell? Embryonic is found in embryos, obviously. Adults are found in adults. The embryonic can make most kinds of cells. Adults are only found in developed tissues and are already partially specialized. I don't know why I put that in there. Does anyone know why sunburns hurt and like you get all dry and like peely after? If you get a really bad sunburn.
Cells are dying.
The cells are dying and the cells are dead. Yes. So which kind of leads us into our last one here is, this apoptosis, that planned cell death. What happens if that doesn't occur? Most of your cells would have the damaged DNA. It then replicates that damaged DNA. You get higher rates of cancer and genetic diseases. So when you get a sunburn, your skin turning red and peeling and falling off is the cells actually dying. And that's programmed. Your body's doing that on purpose. There's too much damage. We want to get rid of these cells.